Hi, the human genome is huge, and we can probably see that the complexity of the organism as humans is so much compared to many other organisms. And there are many other genomes which are larger than even the human genomes. And then what kind of genetic elements are present in there? We'll try to have a look at some of the types of genes that are present and genetic elements present in the human genome. I'm Dr. BCMR. So if we look at the genome in general, we will find that there are several genes, like one is depicted here, there is another one here and another one here. What is a gene? Majority of them code for proteins which are open reading frames with a start codon and then a stop codon. The broken arrows here are the ones that are usually indicated as promoters. They are representative of promoters. The genes themselves are intervened or interrupted by certain regions that we call them as introns. The sequence of the DNA sequences that are resulting in production of a protein are called as exons. So we'll try to see some more of those sequences and these mechanisms. One of the very prominent sequences that are present in majority of the genes is that on eukaryotic genes is that the presence of introns such as these. Prokaryotic organisms do not have introns, and if you see yeast, they have less number of introns. As we increase in complexity, we find that there are more number of introns. We don't know if introns were there and they were lost by bacteria because they need to grow faster, or we don't know if the original genome or the ancestral genome came up without introns and later developed introns. Intron late and intron early. These are the two hypotheses that are prevalent. I think that introns have evolved later, but yes. So upon transcription, exons and introns, both of them get transcribed, and this is called as pre-mRNA. And upon splicing, which would mean the joining of exons, which would imply that the introns are eliminated. So splice means to join, and here the joining is of exons. And once the exons are eliminated, which would mean exons are joined and introns are eliminated, then we call that as mature mRNA. mRNA is then transported into the cytoplasm where the protein is translated, or the mRNA is translated to produce a functional protein. But occasionally what does happen is that the mRNA, mature mRNA, may get reverse transcribed. Reverse transcriptase is an enzyme which is RNA dependent DNA polymerase. It uses this mature mRNA as a template and produces a double-stranded cDNA, complementary DNA of that mRNA. There are several viral elements in our genome. Some of them do encode reverse transcriptase, which may do this activity. And it's a rare occasion, but it does happen. This mRNA, that reverse transcribed or cDNA, the produced double-stranded cDNA, could be integrated into the genome in any of these regions, and if they are in a neutral site, the genome is not going to have detrimental effect, so it would survive. Those sequences are called as pseudogenes. Majority of the time they are not transcribed because there is no promoter. Even if they are transcribed because of any mistakes during copying or reverse transcription, there is a possibility that they would not form. Even if transcribed and translate, they might not produce a functional protein. But they may also, occasionally when it happens, they may cause diseases too. Those regions are called as pseudogenes. In some other cases, assume that there is a gene functional as such, and there could be some mutation. Mutation is change in the sequence, and we are interested about changes in the sequence that have an consequence on the phenotype. Those are mutations and of some importance to us. Most diseases have a genetic underpinning, and they may be because of mutations. At least cancers, all of them are because of mutations. So the mutations can be these changes. Mutation is change in sequence, and change in sequence can happen because of insertion of any DNA in there, or it could be deletion of some parts of it, or it could be substitutions, inversions, duplications, etc. The consequence of the genetic sequence change could be that that could be because it might not be transcribed in the first place. Say, for example, if an insertion happened in the promoter region or regulatory elements, maybe transcription is interrupted. In some other cases, maybe it happened somewhere like this, because of which the protein that might form may be a non-functional protein. Most of the mutations would result in loss of function, but it's not a general rule. There could be a gain of function as well. But in many of the cases, the mutation or the protein that is produced is mutant and is not functional. And there are yet other sequences that are important for the maintenance and stability of the chromosome itself on the, you can say, global scale. Those are telomeres. Telomeres are important to solve the end replication problem. They are important for protection, to provide protection from exonucleases, and most importantly also is that they will prevent any recombination of these ends with ends of other chromosomes through non-homologous end joining. That way they provide a large stability to the whole chromosome. Second point is we need origin of replication. Every genetic material should have origin of replication. There should be some means of the replicating machinery to identify and replicate the DNA. If to define what is a genetic material, I think we should have, this is the primary quality, that it should be able to replicate. There should be mechanisms of replication for it. So in eukaryotes, because they are linear chromosomes and very long DNA, though they need to have multiple origin of replications, usually 40 kb apart from each other. And that is the significance of origin of replication. The next one is the centromere, which is important for segregation of the daughter chromosomes to the two newly formed cells, and without which we might end up with aneuploidy. Some genes may have more, some daughter cells have more number of chromosomes than they should be having. And upon the centromere, there are protein structures that form, which are called as the kinetochores, which are the ones that bind to the microtubules and pull the chromosomes to the opposite poles of the cell. In summary, we can see the human genome, which is 3.2 gigabases, base pairs, and there are gene-related sequences and those elements that are present in the intergenic sequences. This is about 2000 MB. That means there is large amount of space between genomes itself, between genes in a genome. Within the genes and related sequences, we have related are about 1100 MB, and genes are just 48 MB. That means very small number of proteins are actually coding for, a small number of genes are actually coding for proteins. And that is just 48 MB, about 25,000 open reading frames within human genome. The related sequences have these introns. Introns are the ones that we already discussed, intervening sequences. They can be very large, and some of the introns actually also have other genes. Some of them encode RNA genes as well that have very high important regulatory functions. Then there is UTR, which stands for untranslated regions. Say in a mature mRNA, we have something like this. We have this region here and this region here. What is translated? Translation would start in this location and would end here. Start codon and stop codon. The regions here and here are referred to as the untranslated regions. 5' untranslated region and 3' untranslated region. And those are some of the sequences, and the gene fragments are formed because of mutations, and some mutations might make them non-functional, and those are some of the gene fragments. And pseudogenes are the ones that are formed by the reverse transcription of mature mRNA and integration into the genome. In the intergenic region, which is a large chunk of the genome, there are several genes. There are genome-wide repeats which account to 1400 MB. That's huge. And there are other regions such as unique regions, miRNA, microRNA, and other sequences. MicroRNAs do have an important function in the regulation of several genes. If they are manipulated or malfunction of them could cause diseases. And there are also microsatellites, which is about 90 MB. Microsatellites are small regions, say 5-10 bases, and the sequence keeps repeating in that one region for a long time. Those are referred to as the microsatellites. If you look at the way these sequences are distributed within the genome, it seems this is the genes, actual genes coding for proteins is very small compared to the large number of regions that we don't understand or find them non-functional so far. It's not about the number of genes we have, it's all about the regulation of these genes. That's what makes this complexity of brain, movement, and many other things that has allowed us to be successful as a species, but not necessarily as a member of the ecosystem. I hope you got a fair idea and appreciation for the genome and its genetic elements. Thank you. See you later.